cryopreservation is the storage of blood or living tissues at extremely cold temperatures.
Human tissue is stored in liquid nitrogen at or below negative 190 degrees. It is quite costly.
It's costly and not covered by Medicare in Canada, so a lot of people don't use it.
Yeah, and human tissues can be stored for a maximum of 5 years. Section 1, sperm freezing. Rate for without washing is 24.9% and with washing is 35% of living. Process type 1, slow freezing. Takes 2 to 4 hours.
Step 1 is slowly decreasing the temperature of the semen from room temperature until it reaches cold.
Step 2 is adding cryoprotectants, which protects the tissues.
Cryoprotectants are basically like an antifreeze because the cell is mostly made up of water. So to ensure that the cell doesn't explode when it's frozen, they replace the water in the cell with the cryoprotectants, and it's like an antifreeze. Step 3 is putting the samples of semen into a straw. And they just
Semen.
pop the bad boys in there.
They go in.
And then they dip them into the liquid nitrogen where temperatures reach negative 196 degrees Celsius. During the initial cooling off period, the semen is thawed from 5 degrees Celsius to negative 80 degrees before it is put in the semen at a rate of 1 to 10 degrees per minute.
Section 2, rapid freezing of the sperm.
Sperm is mixed with cryoprotectants. The sperm is mixed with cryoprotectants in a straw and left to incubate at 4 degrees for 10 minutes.
Then, hung over liquid nitrogen for 15 minutes. Liquid nitrogen is around negative 80 degrees.
So basically the little guys freeze and they go in ice cubes.
Gets all frozen.
Like such.
Step 3. The straws containing tissues are dunked into liquid nitrogen. Section 2. Embryos freezing. Embryo is the fertilized egg of a human following cleavage.
Since this little guy is full of water, like the sperm here,
Get him out of there.
cryoprotectants are added to replace the water in order to avoid freezing. The embryos are put into a straw, just like the sperm.
Straw. Go in.
And they are put into a freezer.
To freeze.
They're cooled at a rate of 0.3 degrees per minute.
And stored in liquid nitrogen at negative 196 degrees. Frozen embryo, right there.
Cryoprotectants that were added also act as an osmotic gradient, which means that they help water to move across the egg's membrane to avoid freezing.
Section 3, oocytes. This step of freezing is the same as embryo freezing.
An oocyte is a cell of an egg or ovum which develops by meiosis, and it's just essentially an unfertilized egg.
Oocytes have reached a survival rate of 60% after freezing.
For thawing the gametes, the sperm are put into different straws than the ones that they were frozen in and laid up in a warm water bath, and they are gradually warmed up to room temperature.
As they are falling out, they transfer to a smaller container to reduce the toxic effects on the tissues.